Welcome! Today we're setting off on a journey to better understand our ocean, starting with the Essential Ocean Variables and the specification sheets developed by the Global Ocean Observing System, or GOOS. This video will help you understand how to use these tools to help you collect ocean observations in a consistent way so that the data can be compared across nations, regions, and globally. Ocean observation is crucial. Covering 70% of the Earth's surface, the ocean plays an essential role in regulating the climate, providing habitat for a rich diversity of life, and supporting human activities. It facilitates global trade and offers natural resources. For example, more than 3 billion people depend on food resources provided by the ocean. But we cannot effectively manage what we do not understand, and ocean observations helps us understand the ocean to make better decisions on how to protect it and use its resources sustainably. However, measuring the ocean is not a small task. It requires cooperation and must be a collective endeavor if we are to effectively monitor its state. In this context, the Global Ocean Observing System helps us coordinate how we observe the ocean using the Essential Ocean Variables, or EOV framework, as proposed in the Framework for Ocean Observing, to promote common standards for data collection and management and allow comparability and integration of the data. What are EOVs? The GOOS EOVs are key physical, chemical, or biological measurements needed to assess ocean state and variability for important global ocean phenomena, and to provide essential data for applications that support societal benefit. GOOS EOVs are identified to deliver value in three main areas: understanding climate, enabling operational services such as weather forecasts or early warning systems, and keeping track of the health of marine life from local to global scales. Some examples of EOVs include sea surface temperature, nutrients, or seagrass cover and composition. But what makes an EOV essential? First, it must be impactful. Why does it matter? And secondly, it must be feasible to measure. Can we easily collect the data? The purpose of the specification sheets for each EOV is to provide detailed information about the variable, explain its importance, and offer guidance on how best to measure it and how to manage and contribute the data. This figure briefly summarizes each section of the specification sheet, highlighting the value of the information provided for different audiences. Indeed, the specification sheet is a tool that can speak to policy and decision makers, funders, by highlighting the EOV's importance and applications, and inform about the status of the observing system. Second, for observing systems and data collectors, they provide guidance on how to collect observations and what observing approaches can be used, including helpful links to methods and protocols people can consult. For researchers, it provides examples of some of the phenomena, processes, and events that can be observed with this EOV, and the scales at which these observations should be taken. For data managers, it provides step-by-step instructions on how to contribute data and metadata to the global system. Now, let's explore the different sections of a specification sheet using different examples of EOVs to illustrate each point. The first part of the specification sheet provides background and justification. It contains high-level information that is particularly relevant for funders and decision makers. More precisely, this part defines the societal and scientific drivers behind the need to measure each EOV and explains how observations of the EOV helps inform international frameworks such as the Sustainable Development Goals or multilateral agreements like the Global Biodiversity Framework for the Convention on Biological Diversity. As an example, collecting information on the EOV dedicated to mangroves, an ecosystem found in tropical and subtropical coastal regions, will help report against some SDGs, such as the 13th on climate action or the 14th on life below water. But it can also provide information for international agreements, such as the Ramsar Convention on Wetlands of International Importance. For further information on these frameworks, you can click on each icon or see the appendix. This is important information as it showcases the relevance and impact of observing this EOV. This table also explains the links with other types of essential variables, such as the essential biodiversity variables and the essential climate variable. For example, collecting information on the EOV mangroves cover and composition contributes to the marine habitat's essential climate variable, as well as to several essential biodiversity variables, such as ecosystem structure or ecosystem function. Finally, this table also directs you to the BioEco metadata portal, which is a window of observing programs that are collecting BioEco EOVs. There are still some programs that are not yet visible in this portal, and we are requesting observing systems and data collectors to please provide information on existing programs not currently included in the portal so we can add them in. Details on the process are covered in section 5. In the first section, we focus on the core aspects of what it means to measure each EOV. What exactly are we measuring and why does it matter? Let's continue with the example of mangroves. What does it mean to measure mangrove cover and composition? To clarify, the table shows that this includes measurements such as mangrove areal extent, canopy cover, mangrove species composition, and mangrove species zonation. These parameters are what we call subvariables, as they help measure our larger EOV and represent the minimum set of measurements required for this EOV. Another type of measurements included in this table are supporting variables. These are important to provide context and to understand the factors that influence our EOV. For example, while mangrove height as a supporting variable does not directly measure the extent of the mangrove area, it provides valuable information about the health and the growth state of the ecosystem. Another key element of the EOV is the derived products. These are outputs obtained by applying mathematical, physical, or statistical models to the EOV subvariables and supporting variables. For example, mangrove distribution estimates, which can be derived using areal extent and fragmentation. The table of the second section is dedicated to phenomena that can be characterized by EOV subvariables. Here we see, for example, that monitoring mangrove cover and composition helps us understand three phenomena: habitat status and trends, changes in species composition, and carbon storage. This only includes examples, and the list of processes and events that could be captured with this EOV is not exhaustive, but it helps illustrate the types of phenomena or processes that can be observed with this EOV. In the example specification sheet, you can see that each process is described, providing the spatial and temporal extent of the phenomena and the resolutions needed to capture it. Let's focus on the phenomena called habitat status and trends. As mangroves are habitats for a wide range of species, such as fish, mollusks, birds, and even some mammals, their health is directly tied to the well-being of these organisms. Therefore, if the mangrove cover and composition starts to decline due to factors like coastal development and pollution or climate change, it can disrupt the ecosystem and negatively affect species that depend on them for habitat, food, and breeding grounds. Hence, by monitoring mangrove areal extent, canopy cover, species composition and zonation, we can determine the status and trends of the mangrove population to better understand the changes occurring in local biodiversity and predict how these shifts may impact the species that depend on these ecosystems. Section 3, called GOOS Observing Specifications or Requirements, provides guidance for observing systems and data collectors on the temporal and spatial resolutions needed for each subvariable in an optimal observing system for an essential ocean variable. This is, if we wanted to have an observing system for fish abundance and distribution EOV that will provide information to serve different purposes or to observe different phenomena, what would be the minimum resolution of the information required for each of the subvariables, with three different options available as guidance, from minimum to ideal. The higher and more precise the resolutions, the better. For example, for the fish species or species composition subvariable for the fish abundance and distribution EOV, the minimum spatial horizontal resolution suggested is 1 degree grid cells, which means that, as a minimum in a global observing system, having information on species composition every 1 degree would be helpful. Ideally, a higher resolution would be best. However, the information in the table is not intended to be prescriptive. The goal is for the ocean observation community to collectively achieve this objective through collaboration and the sharing of standardized and comparable data. Contributing data to the global system is the most important point. In section 4, information on the different methods and the different technologies that can be used to collect these measurements are provided. For example, for the marine mammal abundance and distribution EOV, 5 of the most common approaches involve line transect surveys, photo ID, or other capture-mark recapture methods, passive acoustic monitoring, biotelemetry, and genetics including environmental DNA. Line transect surveys systematically survey a defined area by following pre-planned transect lines and recording the location and number of marine mammals sighted. Passive acoustic monitoring involves the recording of underwater sounds using a hydrophone and relies on the individual animals vocalizing. It does not produce information where animals are quiet. Biotelemetry is the use of small electronic logging devices attached to individuals to understand their movements, behavior, and/or physiology. Data can be relayed via satellites and mobile networks or downloaded directly if the device is recovered. Genetic sampling, including an emerging capability to sample from seawater samples, that is using environmental or eDNA, can also provide information on species and location. For each approach, the table in the specification sheets provides references to suggested protocols that users can consult to know how to collect these measurements using these approaches. Finally, while ocean data form the backbone of the ocean observation system, the latter wouldn't be effective without adherence to certain commitments regarding data standards and its management. Indeed, the GOOS approach to data management aligns with open data and FAIR principles, that is findable, accessible, interoperable, and reusable, without which we are unable to compare data from different regions and undertake global assessments of biodiversity. To ensure these principles are upheld, each dataset must be accompanied by minimum metadata requirements that allow tracking of the data source, methodologies, and etc. Adding your metadata to the GOOS BioEco portal will be extremely important as the minimum step to take, even if your data is not open. This allows us to know who is collecting data, the type of data, and the location, making it easier to identify gaps in observations. The BioEco portal provides you a window to biological and ecosystem ocean observing. Metadata information can be crucial. For instance, in emergencies like an oil spill, if we have knowledge of people that collected information in that location before the event to assess its effects. The final section provides the procedures for contributing data to the global ocean observing system, and specifically to the BioEco metadata portal, and to the Ocean Biodiversity and Information System OBIS. This will take you through a step-by-step process to either submit your data directly to OBIS or connect your data center with OBIS. More details and helpful resources are available in this section, including contact points through OBIS nodes and links. Additionally, for some EOVs, this section also directs you to existing useful data products. This is the end of this video. Thank you for your attention, and we hope that this has helped you understand how to collect ocean observations in a consistent way. More resources and helpful links are provided in the description of this video.